Plastic pollution in the marine environment has become a real global issue. Most common plastics can't be broken down by nature and can thus be found in the ecosystems, for example on Texel. One of the many plants that can be found in one of these ecosystems, the salt marshes of Texel, is Salicornia spp. This plant has to deal with all kinds of stressors, amongst these is plastic. Some of these stressors have already been researched, but plastic as a stressor in relation to Salicornia hasn't yet. Thus we wonder, what is the effect of macroplastic smothering on the growth and survival of Salicornia spp? We hypothesize that plastic smothering of Salicornia spp is negatively correlated with the growth rate and positively correlated with the mortality rate.
For the lab part of our research, we took 24 samples of Salicornia spp from the same patch in the slufter, ensuring they were seedlings at similar stages of development. They were cut out of the ground with tubes containing drainage holes to limit root damage and keep the substrate composition similar. Plants that struggled before the study were excluded from statistical analysis. The samples were placed in a tub where we recreated the tides by adding seawater and taking this out after 2 hours, twice a day. A bubbler helped creating a continuous flow of water. To experiment with plastic smothering, we covered 8 plants with closed tubes and 8 with open tubes made from black trash bags that were taped to 2 small wooden sticks in the soil. We used a growth lamp to promote growth and a rotation screen to ensure consistent conditions. We assessed plant growth and survival using various methods, including measuring length and color on days 1 and 4 and determining the fresh and dry weight after day 4. In the field research, we examined cover and death among Salicornia plants by comparing plots with and without algae smothering in the slufter. To assess the survival of plants during the field research, we determined the amount of plants in smothered and unsmothered plots and determined the angle of certain amounts of plants in each plot. To analyze all the data we obtained, we made use of a t-test for the field research and an ANOVA test for the lab research.
And these are our results. Firstly, for the lab experiment, this graph shows the average difference in length between each type of plastic treatment. Heavier plastic treatment shows more growth per day. And the p-value is lower than 0.05, which indicates a significant difference between groups. But the Tukey test specified that the difference in length is only significant between the heavy plastic and the control treatment. This graph shows the average weight difference between wet and dry weight for each type of plastic treatment. Again, heavier plastic treatment shows a larger difference than the medium or control treatment. However, this difference between groups is not significant, as the p-value is larger than 0.05. Moving on to the field experiment, we can see that the overall density of Salicornia is higher in the smothered plots, but the p-value is larger than 0.05, and so this difference is not significant. We also derived that the number of healthy, unsmothered plants is higher in unsmothered plots, and this difference is significant, since the p-value is lower than 0.05. In this last graph, we can see that the average angle is nearly the same for both plot types. Statistical analysis confirms this. The p-value is higher than 0.05, and so there's no significant difference.
This research, of course, has some weaknesses. For example, for the lab experiment, not all plants reached the top of the tubes, so we had to add sand to the bottom and push the sediment up, leading to changed and differing soil compositions, which might have affected the oxygen levels and water retention and thus growth. Furthermore, some plants, especially those without plastic cover, dried out on the second day due to the heat in the tent, which stressed them. Lastly, green parts broke off of 2 plants while measuring, reducing their weight and impacting the results. Further research can be done on the impact of other types of plastic and should also extend the growth period and investigate the impact of soil composition and compression. Some assumptions were made for the field experiment. Firstly, that the smothered Salicornia has died as a result of algae smothering, and secondly, that the current angle is the same angle as the moment of death. If these assumptions are incorrect, then the results are invalid, so further research should examine this. Overall, we see that certain plastics might temporarily benefit Salicornia, but we doubt that this will be the norm, as our research was conservative and not all-inclusive. So we expect Salicornia populations to decline with increased plastic amounts, which is troublesome as Salicornia is a pioneer species that shapes its environment and can be used as a food source.
All in all, in the short term, plastic smothering seems to increase the growth of Salicornia spp, and it doesn't seem to have an effect on the survival rate. The long-term effects are, however, still unknown and might differ.